Hello, my name is Haley Coolset, and I'm going to be doing this video on chemoreception. So what is chemoreception? So chemoreception is going to be the recognition of chemical stimuli. So the specific chemical stimuli that we're going to be covering today is going to be smell and taste. So chemoreception is going to be a process within the peripheral nervous system, and more specifically we're going to be looking at the afferent sensory portion. So these signals are going to start within the periphery, and then they're going to be sent back towards the central nervous system. So chemoreception and olfaction specifically is one of the oldest known senses, and all sensory pathways actually have these 4 different steps in common. So not just what we're going to be talking about today, but you can apply these 4 steps to any other processes within the sensory system. So reception, you are going to have some sort of receptors that are going to detect the stimulus. Transduction, so that's where you'll have your change in a membrane potential that will end up going over the threshold and firing off an action potential. Transmission is going to be the way in which the neurons take the action potential towards the central nervous system, and perception is going to be when that signal makes its way to the central nervous system. That's going to be how our brain interprets and perceives everything. So we'll start off with olfaction, or smell. So olfaction is going to allow us to differentiate between hundreds of different types of odors. And what's important to know about olfaction that makes it different from the other sensory processes is that this is the only sensory pathway that we will not find synapsing within the thalamus in the brain. So we're actually going to start out with this slide. So this is the olfactory epithelium. This is going to be just below the bone, and you'll see that on the next slide with a better view of the overall anatomy. So we have Bowman's gland here in the center. We have our sensory neurons, and we can actually take a look from the bottom up. So we have the mucus layer. That's where that is where the chemicals and the smells and the odor molecules, they are going to dissolve within this mucus layer. They get dissolved. These are the dendrites, or the olfactory cilia. They're going to take up those odor molecules through our olfactory sensory neuron, and then it's going to take it out the top here through the olfactory neuron axons, which is going to take the information to the olfactory bulb. So on this next slide, this gives us a better view of everything that we're looking at from the outside. So starting at the bottom, this is a smaller view of what we were just looking at. So that's the olfactory epithelium. So we just had our signals taken up from there. They're going from the olfactory epithelium. They're driving down that pathway through the bone, and they're actually going to synapse here in the olfactory bulb. Once they synapse in the olfactory bulb, our secondary sensory neurons are going to take these signals down the olfactory tract into the olfactory cortex, and then those will go to the cerebral cortex and also the limbic system. So that is where we are going to have our perception of what we just smelled. So what's important to know is that the cranial nerve that we're going to be using for the olfactory pathway is going to be cranial nerve number 1. So now we're going into gustation. What is that? Taste. So taste is actually going to be very closely linked to olfaction. So if you think about when you ever had a cold and all of a sudden you can't smell and now you can't taste. So these are 2 parts of chemoreception that are going to be very similar. So gustation is going to be a combination of 5 different sensations. This is what they've come up with so far. They think that it could potentially be more. So we have sweet, salty, bitter, sour, and umami. They're going to be the 5 different taste sensations. And each one of those sensations is going to be linked with a specific physiological process. So we'll take a look at this slide from Silverthorne. Up in the top left, we have a tongue. On the tongue, we have the taste buds. So the taste buds are going to be where we have those receptors to start this process. Just below that is a light micrograph of a taste bud. And at the top of the taste bud, you actually have a taste pore, and that's going to be the little entrance to the dorsal side of the tongue. On the right-hand side, this is going to show us our 5 different types of tastes and the receptor pathways that they very briefly describes what they're going to go through. So, sweet, umami, bitter, sour, and then we have salt over on the right-hand side. So let's go into that a little bit deeper. So this is going to be taste transduction. So we're going to have 2 different pathways. So we are going to have the pathways where the ligand is going to bind to a G protein coupled membrane receptor. And then we have the other pathway where it is going to bind with a specific ion channel. So we'll start with the ion channels over on the right, and that's going to be responsible for sour and salty taste. So what happens, you're going to have your ligand come around. In this instance, we'll go through sour. The hydrogen cell actually is going to go through this ion channel, and that's going to stimulate calcium to come into the cell. That's going to stimulate exocytosis. So you'll see here, calcium comes in. Within this synaptic vesicle, we are going to have neurotransmitter. In this case, it's typically serotonin. Comes down to the bottom where it's going to release the neurotransmitter onto our primary gustatory neuron. Similar thing would happen for when you eat a salty food, except in that instance, sodium is going to be the ligand that's going to do all the work. On the other half, we have our G protein coupled receptors. So this is going to be sweet, umami, and bitter. So what's going to happen is your ligands are going to bind and they're going to activate your taste cell. That's going to stimulate this protein Gustation, and then your signal transduction is going to begin. So in this instance, we're actually not going to be using we will be using, so calcium is going to come into the cell. It's going to trigger ATP formation instead of exocytosis. So we come down to the bottom and it's actually ATP that's going to be released, and that is going to be picked up by the primary gustatory neurons. So once our primary gustatory neurons have been activated, those axons are going to travel down through cranial nerves 7, 9, and 10. They are going to synapse within the medulla. From there, the axons are going to travel up into the thalamus, and from the thalamus, those are going to travel to the gustatory cortex, which is where we are going to get our perception of taste. So here's my reference from Silverthorne. And then this is going to be, so this is the example of the additional study module that I did. So this is actually filled out, and I'll attach that to the video when I submit it for you.